Welcome students. Now we are going to see enzymes involved in DNA replication. What are the enzymes are going to involved in the DNA replication? Okay. Next, the introduction. A fundamental property of living organisms is their ability to reproduce. Organisms produce offspring that are similar to themselves. Okay, it means that every organisms can be able to reproduce their daughter cells. Okay, the process occur by which a single parental cells divides to produce two identical daughter cells. Okay, the single parental cells want to divide into to produce the two daughter cells. Okay, in this process of genetic material must be duplicated prior to cell division so that the daughter cells receive the full complement of the genetic information. So see, when the daughter cells are forming, it means that parental cells are divided into daughter cells. In that case, the daughter cells want to get the full complete genetic information to their parent cells. Okay, then next one is another replication. Replication means the synthesis of daughter nucleic acid molecules identical to the parental nucleic acid. That is a replication. Okay, the synthesis of daughter nucleic acid molecules identical to the parental nucleic acids. Thus, accurate and complete replications of the DNA is essential to the ability of cell organisms to reproduce. It is the basis for biological inheritance. Okay, DNA is made up of double helix of two complementary strands. That is the DNA. See, do you know the DNA? DNA, that is the double helix structure. There have two strands. Okay, during the replication, these strands are separated. Then that two strands are separated into a single strand. Then they are going to the replication process. Then what are the enzymes are involved in the DNA replication? See, above the slides are indicating what is the replication. Then now we are going to see what are the enzymes are involved in the replication. DNA Helicase, DNA polymerase, DNA clamp, single strand binding proteins, Topoisomerase, and DNA Ligase and Primase. These are the enzymes are involved in the DNA replications. Okay. One by one, we are going to see what are the enzymes and what is the role of the enzymes. First, DNA helicase. Helicase were discovered in the year of E. coli in the year of 1976 and are class of enzymes vital to all living organisms. Okay, this is in the year of 1976. Also known as helical destabilizing enzyme. What is it known? That is, DNA helicase is a destabilizing enzyme. They separate the two strands of DNA at the replication fork behind the topoisomerase. Okay, another one enzyme is in a topoisomerase. I will tell you about. I will tell you later to that what is a topoisomerase. Now we are going to see the helicase. Okay, helicase means that is a destabilizing enzyme. It means the double-stranded DNA is converted into a single-stranded DNA. See, when that enzyme is function, and that case, that the double-stranded DNA is split into a single-stranded DNA. After that, the topoisomerase is used to strengthening to that DNA strand. They are motor proteins that move directly along the nucleic acid phosphodiester backbone, separating two unrelated nucleic acid strands. That is DNA, RNA or DNA RNA hybrid using energy derived from the ATP hydrolysis. They have their molecular weight is 3 lakhs, which contains 6 identical subunits. I will give it to you later. See, this is the structure of this double-stranded DNA is converted into a single-stranded DNA. When the helicase is functioning, it's look like a zip model. Okay, when the zip is closed and zip is open. When the zip is closed and the two strands are joined to that one, then zip is open in the sense that the two single strands is in there. So when it's bind to the helicase, the single-stranded is unwinded. Okay, then this is DNA polymerase. Okay, DNA polymerase is an enzyme that synthesizes the DNA molecules from deoxyribonucleotides, the building blocks of DNA. These enzymes are essential to DNA replication and usually works in pair to create two identical DNA strands from the single original DNA molecules. During this process, DNA polymerase reads the existing DNA strand to create two new strands and match the existing one. Okay, this is repair corrections also. DNA polymerase enzyme to use to synthesize the DNA molecules. Okay, what is the role of the DNA polymerase? Used to synthesize the DNA molecule from the deoxyribonucleosides. Okay, this is the DNA polymerase. DNA polymerase, when it's bind to the DNA polymerase, that it's lead to synthesis of new DNA strands. Okay, then now first the DNA strand is separated, then it synthesis the newly synthesis of DNA strand. These enzymes catalyze the following chemical reaction, that is deoxynucleotide triphosphate plus DNA gives a diphosphate DNA. Okay, diphosphate DNA. Catalyze the DNA template directed extension of the 3' end of the DNA strands by one nucleotide at a time. 3'. Okay, it starts to the 3'. The known DNA polymerase have highly conserved structures. Okay, this is how a very conservative one. The shape can be described and reassembling the right hand with the fingers to the domains. DNA polymerase can be subdivided into different families. Okay, then the families are prokaryotics and eukaryotics. Okay, that DNA polymerase are different from the eukaryotics and prokaryotics. Eukaryotics that are in DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4 and then DNA polymerase 5. Okay, in a eukaryotic, there are more than 15 DNA polymerases in that. Then DNA polymerase alpha, sigma and gamma is written here. Delta, sorry. Then DNA polymerase epsilon is also that eukaryotics. These are the families of eukaryotics. Then the prokaryotic DNA polymerases, what is the activity of that one? Okay, the DNA polymerase 1, it used to cap filling of cap after removal of RNA primer or DNA repair, removal of RNA primer. Okay, that is 3' to 5' or 5' to 3'. Used to cap. See, when it's formed to that gap, that want to fill it. Then DNA polymerase 2, it is a DNA repair to the direction of 3' to 5'. Then DNA polymerase 3, that is the main enzyme of replication. Okay, that is that replication, proofreading and editing from the 3' to 5'. All the replications, proofreading and editing, entire things are only carried out by the DNA polymerase 3. This is the main enzyme of replication. Okay, next eukaryotic. Okay, alpha, beta, gamma, delta. Okay, this is delta is important to that one replications of DNA. Okay, then alpha replication of DNA, that is exclusions, exonuclease activity is noticed in that. At least 9 polymerase is known as a major replicative enzymes. Okay, let's see that how only 9 polymerase enzymes Y stands that polymerase delta that have major replicative enzyme is done that. Okay, then DNA clamp. The DNA clamp, also known as a sliding clamp, as a protein fold that serve as a preservativity promoting factors in DNA replication. The two is a sliding clamp. Okay, like that clamp. Do you know the clamp? Okay, then as a critical component of our DNA polymerase 3 hollow enzyme, the clamp protein binds to the DNA polymerase and prevent this enzyme from dissociating from the template's DNA strand. Okay, see, when the clamp is formed, that it is indicating it is used to dissociate the DNA template strand is to prevent that enzymes. Clamp polymerase, the protein-protein interactions are stronger or more specific than the directions to that one. Okay, the template strand is used to when the clamp is formed, the protein-protein interaction is very stronger to the DNA template strand. Okay, see, when it's bind to this is the structure of DNA polymerase. When its clamp is formed, it is used to do very strong to the protein-protein interactions to that one. Single stranded binding proteins. Next, we are going to see that this SSP proteins. Okay, single stranded binding proteins is to prevent the re-annealing. That is, the binding of complementary DNA sequences to protect the single stranded DNA from the being digested by the nucleases to prevent the secondary structure formation, thereby allowing other enzymes to function effectively to the single stranded. See, when the double strands are formed, after that, what is the role of proteins? The proteins want to change primary secondary structures. Okay, that in that case, the protein want to be folded to that one. So they want to prevent it. When it's bind to the single stranded binding proteins to that one, it used to prevent the folding to that DNA strands. Okay, then after that, it used to further enzymes or effectively binded to the single strand. Then molecular weight of SSP protein, that is 75,600 tolls of molecular weight. Okay, that is, it contains 4 identical subunits which bind to the single stranded DNA. They have 4 different synthetics. That is, when it's bind to the single stranded binding, it used to unwinding to that one, stability to that one. Then topoisomerase. Okay, this is an enzyme, and this enzyme is used to, that is an unwinding process. Every cell has an enzyme that increase or decrease the extent of DNA unwinding are called topoisomerases. Topoisomerase is also known as DNA high-rise and DNA high-rise, and that act as a topology of DNA. Topoisomerase is also known as DNA high-rise. Another way is called as a DNA high-rise. Okay, what is the role of topoisomerase? It is used to DNA unwinding process. Topoisomerases bind to double-stranded DNA and cut the phosphate backbone to either one or both of the DNA strands. This intermediate break allows the DNA to be untangled, okay, untangled or unwound, and at the end of this process, the DNA backbone is released again. Okay, that is linking number. What is that linking number? That is a DNA polymerase used to unwinding that process to that one. Okay, linking number is a topological property. It can be defined as the number of times on second strand previous the second strand surface. Okay, that is indicating the linking number. How many times are topological properties are doing to that one? That is indicating a linking numbers. That is 2. That is type 1 topoisomerase and type 2 topoisomerase. Type 1 topoisomerase, this act by the transitional breaking one of the 2 DNA strands, rotating one of the ends about the unbroken strand and rejoining the broken ends. They change linking number in the increment of 1. Okay, that is once enter into the first time in breaking to that one, that is 1, and then second one, that is increments that are breaking to the number 2. Okay, that is next one we are going to see DNA ligases. This is a specific type of enzyme, a ligase, that facilitate the joining of DNA strands together by catalyzing the formation of the phosphodiester bonds. Okay, DNA ligase, that is an enzyme, and that enzyme used to joining of DNA fragments. Okay, DNA ligase is used in both DNA repair and DNA replication. The mechanism of DNA ligase is a form of 2 covalent phosphodiester bonds between the 3' hydroxyl end of the 1 nucleotides with a 5' phosphate and another one, that is an acceptor and donor. ATP is involved in the process of that is 3 steps is involved in the DNA ligases. First, adenylation, okay, transfer, and then formation to that one. Adenylation, addition of AMP of glycine reduced in the active center of the enzyme pyrophosphate is released. Then transfer of the AMP to the 5' phosphate of this called donor. And next one is an acceptor. Formation of phosphodiester bond between the 5' phosphates of the donor and the 3' hydroxyl of the acceptor. Primer. There is a primase. This is an enzyme is involved in the replications of DNA and in a type of RNA polymerase. Primase catalyzes the synthesis of short DNA segments called the primer complementary to the single-stranded DNA template. Okay, that is a RNA polymerase. Okay, primase to initiate the DNA. Primase is a key importance in DNA replication because no non-replicative DNA polymerases can initiate the synthesis of DNA strand without an initial RNA or DNA primer. That is a temporary DNA elongation. After the elongation, the RNA piece is removed by the 5' to 3' exonuclease and refilled with the DNA. See, when it's bind to the DNA polymerases enzyme, that replication process is involved. Okay, when it's bind to the DNA polymerase to the TNU1, then RNA polymerase is bind to that. When it's bind to the DNA primase, then it's bind to the bind to the RNA polymerase. Then process is initiated. After the elongation process, that RNA polymerase is removed and filled with the DNA nucleotide base pairs. Okay, that is the directions of replications. See, when it's bind to the primase, when RNA primer is binded to that one, then it's initiated, elongated to that one process. Okay, this is the entire things of topoisomerase. What are the enzymes are involved in that one? Topoisomerase, RNA polymerase, RNA primase, DNA ligase, helicase. Okay, single stranded binding proteins. These are the enzymes are involved in the DNA replication process. Thank you to all.